Another day, another story. Luminous species are widely scattered taxonomically, with no discernible pattern. Many luminous shrimps are known but no luminous crabs. Welcome to Tabo Eminent Channel. Many luminous squids are known but only a single luminous octopus, Calliced arachawai of Japan. Again, luminous centipedes and millipedes are not uncommon, but luminous scorpions and spiders are apparently non-existent. Almost half the animal phyla contain luminous forms, but the number of representatives is very small compared with the total number of known animal species. The protists are not so rich in luminous species but are greatest in sheer abundance, especially in tropical seas. In fact, the majority of luminous organisms are marine. The ocean surface in many parts of the tropics is dense with single-celled luminous planktonic organisms, primarily dinoflagellates, that glow when stimulated mechanically, as by the churning of the waves, or, when washed ashore, by the pressure of a foot. Some organisms exhibit a 24-hour rhythm of light intensity, highest at night and lowest during the day. Among crustaceans, luminous species are especially remarkable in the copepods, shrimps, and ostracods. Luminous copepods are widely distributed throughout the world's waters. Some are surface dwellers, while others live in the deep sea. Two famous groups of luminous copepods are Pleuroma and Metridia. Some shrimps, Hoplophorus, emit a luminous secretion from luminous organs, while others possess true light organs, photophores, which consist of a lens, reflector, and light-emitting photogenic cells. Of the 3 or 4 species of the ostracod genus Vargula, also known as Cypridina, known to be luminous, the most famous is V. hilgendorfii, or C. hilgendorfii, found in the coastal waters and sands of Japan. That tiny, shelled organism, which ejects a blue luminous secretion into the water when disturbed, may be collected and dried for the light-emitting components, which are active indefinitely. Water jelly, Equoria victoria. The medusa stage of the bioluminescent water jelly or crystal jelly, Equoria victoria, in the waters off the west coast of North America. The species is harvested for its luminescent protein called equorin, which has been used in medical research as a fluorescent marker protein. More. Other organisms responsible for large patches of light in the ocean are jellyfish and other coelenterates and comb jellies, ctenophores. A large proportion of the floating, transparent siphonophores and the feathery, bottom-dwelling sea pens are luminous. Many of the hydroids and jellyfish are also luminous. Sea pens, Pentula, sea cactus, Cavenularia, and sea pansy, Ranilla, are colonies, which upon stimulation generate a wave of luminous light that travels down the organism. The luminescence in these organisms appears to be under nervous control. Among annelids, marine worms and earthworms both contain luminous forms. Odontocilus, the fireworm of Bermuda, swarms in great numbers a few days after the full moon. Female worms, about 2 centimeters, almost 1 inch, in length, rise to the surface shortly after sunset and swim in circles while ejecting a luminous secretion. Smaller male worms swim to where the females are circling and mate. The male is also luminous, but the light is intermittent and of intracellular character. It is not certain whether luminescence has any relationship to mating, since non-luminous Odontocilus exhibits similar courtship behavior. Catopterus spends its life in a tube of parchment membrane, with openings at both ends. It luminesces when disturbed, but it is doubtful whether the luminescence has any special purpose. Pollino and Polycirrus are luminous annelids that usually live in sand or rock. Luminous mollusks include pholas, a bivalve, Philero, a floating nudibranch, Planaxis, a marine gastropod, Lotia, a freshwater limpet, and squids, cephalopods. Deep-sea anglerfish, hatchetfish, and lanternfish are among the best-known luminescent fishes. In most such fishes, luminescence is produced intracellularly. The light is emitted by special cells called photocytes. The anatomical structure of the luminous organs of many fishes is similar to that of squids. Deep-sea fishes have photophores along the body, under the eyes, and often on barbels or antennae. The typical luminous organ consists of a lens, luminous body, color filter, and reflector. The light is often under the fish's nervous control, and after death the ability to luminesce disappears rapidly. Whether the light-producing components are developed by the fish or ingested by the fish is not clear. A distinct possibility exists that a fish feeds on crustaceans such as vagular organisms and uses their light-emitting components for its own light production. Biochemical events of light emission. In most bioluminescent organisms, the essential light-emitting components are the oxidizable organic molecule luciferin and the enzyme luciferase, which are specific for different organisms. The present custom is to use generic names according to origin, for example, firefly luciferin and luciferase, vagular luciferin and luciferase. The luciferin-luciferase reaction is actually an enzyme-substrate reaction in which luciferin, the substrate, is oxidized by molecular oxygen, the reaction being catalyzed by the enzyme luciferase, with the consequent emission of light. The light emission continues until all the luciferin is oxidized. That type of reaction is found in fireflies, Vagula, Lotia, and many types of fish, such as lanternfish, hatchetfish, apagon, and parapryanthus. In firefly luminescence, the substance adenosine triphosphate, ATP, initially reacts with firefly luciferase, ionic magnesium, and firefly luciferin to form a complex, luciferase luciferyl adenylate, and pyrophosphate. That complex then reacts with molecular oxygen to emit light. Enough energy is liberated in the last step to convert the electronic configuration of the luciferase luciferyl adenylate complex from a low-energy ground state to a high-energy excited state. Luminescent bacteria employ the enzymatic oxidation of reduced flavin mononucleotide, FMNH2. In the complete reaction, bacterial luciferase reacts with FMNH2 and oxygen to form a long-lived intermediate complex, which then reacts with a long-chain aliphatic aldehyde molecule, for example, decanal, to emit light. The significance of bioluminescence in research. The luminescent reaction of the firefly has been used as an assay method for the determination of adenosine triphosphate, ATP, an important metabolic substance used by all living cells in numerous reactions in which energy is either stored or expended. The glow of a specially blended extract of firefly lanterns eventually dims and disappears as ATP is broken down. The addition of fresh ATP, either as a pure chemical or as a constituent of a tissue extract, immediately restores the luminescence. The intensity of the glow is a direct measure of the amount of ATP present in the extract. That assay method has been widely used in medical and biological research to determine the amount of ATP present in extracts of cells and tissues. The study of reactions involving ATP has led to a detailed understanding of the mechanisms of energy conversion in cells. The firefly reaction is one of the few reactions in which ATP is directly involved with light emission. All other bioluminescent reactions involve compounds that are chemically distinct from ATP. Thanks for watching. Request you to subscribe the channel.